Good morning and greetings from the New York Botanical Garden. My name is Jennifer Bernstein, and it's my honor to serve as the CEO and the William C. Steer Senior President of this distinguished organization. It's a pleasure to welcome you today on International Women's Day during Women's History Month to this special webinar to celebrate the life and legacy of Carol Gracie, a woman who was an accomplished and beloved member of the NYBG community for more than 4 decades. Women have played pivotal roles in NYBG from this institution's very founding. For example, this year during Women's History Month, in addition to this webinar, another offering that we have online is a continuing education course beginning tomorrow, March 9th, about 3 pioneering 20th century women garden designers, Beatrix Ferrand, Marion Coffin, and Ellen Shipman. Students in this course can view original plans of NYBG gardens that these women designed, the Peggy Rockefeller Rose Garden, the Benenson Ornamental Conifers, and the Ladies' Border. I mention these designers because their legacies, like that of Carol Gracie, embody the theme of the 2023 Women's History Month, which is celebrating women who tell our stories. Through their inspired designs, these women told stories of beauty, diversity, and tranquility evoked by plants at NYBG and beyond. We subtitled today's presentation, Interpreter of Nature's Stories, because Carol Gracie used her creative talents as a keen nature observer, photographer, writer, and lecturer to tell the stories of plants, animals, and fungi to people of all ages. Thank you for joining us today to celebrate Carol Gracie's life and legacy. And now I'll turn the program over to Dr. Jacqueline Kalunke, Curator Emerita of NYBG's William and Linda Steer Herbarium, who will introduce our speakers. Jackie?
I'm here.
We can hear you.
Yeah, okay, I was waiting for, oh, okay. Well, thank you, Jennifer. I'm very happy to be here to celebrate Carol Gracie's life. I met Carol when she was working in the education department, and I started in the herbarium, and our friendship lasted the next 40 years. She had so many friends. I imagine that most of the people attending this event are also her friends, from French Guiana, from the garden, from her eco tours and lectures, from her walking group, her family, and her neighborhood. We met her in different contexts, but I'm sure we each saw the same person: strong, principled, industrious, and organized, and also loving, supportive, sociable, and funny. We saw a person worth celebrating. Today, you will hear only from 5 of that multitude who found her to be an absolutely lovely person. Before we start the program, I invite audience members to use the Zoom Q&A feature to submit your own memories of Carol. You can do this during the program itself or during a 20-minute period following the end of this program. Our first speaker, Margaret Roach, has been an avid gardener and a leading garden writer for more than 30 years, most notably at Martha Stewart Living and Newsday. In 2007, she abandoned the city and the corporate world to live in her garden in Columbia County, New York, and pursue a more creative life. Since April 2020, she has been the garden columnist for the New York Times, where she actually began her journalism career. Through her 3 books, her website, awaytogarden.com, and her weekly podcast of the same name on public radio, she offers both practical advice and inspirational reflections about gardening. Through her podcast, she has shared conversations with Carol about the natural history of wildflowers. Margaret, welcome.
Thank you. Thanks very much. Here we go. Though I confess I've never read a single mystery novel, I feel I can be absolutely emphatic in my declaration that Carol was one of the great mystery writers of all time. No hidden detail was going to stay that way as she methodically unraveled one whodunit after another for her readers and for audiences who attended one of her lectures or had the privilege of accompanying her on a wildflower walk, as I'm grateful to have done various times. I may not read mysteries, but I do watch birds, and I have for decades. So it really stuck with me when I first heard Carol challenging her audience that when we observe wildflowers, we should think and act like the most expert bird watchers do with their avian subjects. They don't merely make a life list, check, 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 as if it were a competition, and then hurry off to find the next species to add to their tally, but they go about bird watching with the emphasis on watching. Why is that the key? I asked her. Next slide, please. It's not just checking off that you've seen a Canada lily or you've seen a house sparrow, she said. It's really observing what their organism is doing. And there are still things to be learned about even our local wildflowers that have not been observed. Because people just think, well, they're common, they've been here forever, anything that is known about them must be written already. Yet if you take the time and patience to sit there and watch, she added, you discover something new, and you'll definitely discover something interesting. Spoken like the natural, lifelong student and teacher that she was. Carol knew that the secret to getting the big picture was to go small and watch and wait for every subtle detail to reveal itself, and she did, often through the macro lens of her camera. No aspect was too tiny or presumed insignificant. In keeping with that philosophy of hers that discovery was always possible, Carol was an exceptionally patient observer, willing to crouch and crawl around and just wait. And not merely at the moment of peak bloom either, but even at times in the life of the plants when the casual observer might look at them and say, there's nothing happening here right now. Oh, really? Look again and again. Next slide, please. If you let Carol show you the way, you might be privileged to see an act of myrmecochory, the ancient mutualism between ants and early spring ephemerals whose seeds have nutrient-rich appendages called eliosomes, including natives like bloodroot, trillium, Dutchman's breeches, marybells, twinleaf, and even wild violets. She'd explain that the ants don't eat and run, that they carry the seed back to their nest with its lipid-rich body attached, where the eliosome is finally consumed, but the seed is left intact, essentially sown in a spot rich with ant debris, the start of a new planting at a distance from the parent plant. It's a backup in case something befalls the one it came from, and one of the pillars of species survival for as many as 35% of herbaceous understory plants in eastern North America, for whom ant assistance is part of their master plan. You learn all that just from happening on a clump of violets alongside the path during one of your walks with Carol. She would be as patient explaining it all to a student, a reader, a friend, as she had been all those countless times she'd sat watching for the moment when this ancient choreographed ant dance takes place. Next slide, please. While you were humbly on your knees among the violets, she'd tell you about another of their secrets to survival, their backup set of flowers, in case the weather or some herbivorous animal trashed the showy ones dancing up above and among the leaves. These second, more rudimentary blooms, called the cleistogamous ones, are nearly hidden down by the base of the plant, and they don't open. They're capable of self-fertilization, ensuring that seed is set no matter what happens. Next slide, please. She'd urge you to look at milkweeds closely too, and at different times of the season, not just when the adult monarchs might be nectaring at their flowers, and not just for the various life stages of the well-known monarch-milkweed relationship at all, but for the guild of insects who feed on different parts of different Asclepias species. Butterflies and moths, yes, but also beetles, including weevils and true bugs and aphids, and her favorite uncommon milkweed, the four-eyed milkweed, beetle, aka the red milkweed beetle. It's one really curious looking insect, and through that macro lens of hers, she could see how the socket of each antenna on its head divides each of its compound eyes in two, resulting in 2 upper eyes and 2 lower ones. Again, it's the little things. She passed down each aha, explaining all the unfamiliar words and showing you just where to look, always doing it with delight, even though you know she'd probably seen most of it before, like many, many times. One email I got in early spring 2019 from Carol said this: I know that you like learning new botanical terms like marcescent, she said, remembering one of my favorites for deciduous trees whose faded leaves hang on through the winter like some oaks and beeches. Me too, she continued, and I just learned a new one this morning in preparation for beginning a chapter on chicory. Matutinal, meaning of the morning. Chicory blooms early in the morning, she explained, and closes later in the day. And then her email went on, I just told Scott that he is a matutinal person, often pre-matutinal, she wrote. I tell you these details not to teach you anything the way Carol taught me. Most of you listening already know these bits and much, much more than I ever will. But I recount these little plant lessons because that to me was the spirit of Carol, to see, to study, and to share. And so I'm just in my way carrying on that lineage a little bit, that mission here and in my writing. If she were here, she would be excitedly telling us about her latest sightings or some new layer of insight she'd had about some past observation as she kept going ever deeper, always with that enthusiasm. Or maybe she'd just be telling us about a moment of home garden awe. I would get these periodic emails over the years, gardener to gardener, about what was blooming in her yard or about how some plant I'd shared with her was faring. I remember when she told me excitedly about 10 years ago that the May apple in her garden had moved 30 feet uphill from her original patch. Usually this plant spreads vegetatively, she explained, expanding sideways underground. But like those ants and those other spring ephemerals, it's known to have an animal seed dispersal agent too. Next slide, please. Except this time we're not talking ants, but box turtles who eat the seed-containing fruit when perfectly ripe and slowly digest it and then wander off and eventually excrete the seeds. She said that she hadn't seen the turtle yet, but you just know she was keeping lookout, vigilant as ever. In witnessing the aspects of plants' life histories, Carol never appeared to lose the sense of awe, the openness of beginner's mind, despite all her expertise. And even so, I said to her, even as many times as you've seen and lectured and written about some of these species? No, the wonder remains, she said, and then she shared part of the reason why. I often wonder how long it took for that particular evolution to occur, she said. Next slide, please. Carol Gracie was so good she could even solve a botanical mystery without seeing the plant. That was how well she knew the plant communities that she devoted herself to the study of, how tuned in. One June, all I had to say was that I'd seen something new to me in a field that was low growing with flowers like bee balm, but orangey and yellow, and she knew. I was 2 words into my next sentence about to tell her what I'd keyed it out to be when she interrupted me. I know what you're talking about, she said, and she did. Pedicularis canadensis, sometimes called lousewort or wood betony. Talk about oddball life histories. This one is a hemiparasite, she explained, adding another word to my vocabulary, thank you, with chlorophyll in its leaves, meaning it's able to make its own carbohydrates, but it has very few root hairs, so it needs to parasitize other plants to access water and minerals. I could list 100 more things that she shared that stuck with me, but my point is this. I just want to thank Carol here aloud and in the presence of others she may have similarly impacted with her openness to taking it all in and her infectious, eye-opening vantage point. I want to say how grateful I am for the little awakening she provided me over and again. And if you've not yet looked at native plants through her eyes or the lens of her camera, Carol's take on the natural world lives on in the books she left for us. Next slide, please. One last big picture thought from Carol that she impressed on me a few years ago when I asked her why her spring and summer Northeastern wildflower books each has the phrase "a natural history" as its subtitle. The natural history part is what role these plants play in their environment, she said. I think almost anybody can appreciate their beauty, but they're not just there for us to look at, but to play an important role for their pollinators or their dispersal agents. They have a very particular niche they fill, especially in our Northeast forests. So most of all, that's the guidance that I took away from Carol and from her work, that we should look for all the pieces, however tiny, and never stop looking and never stop wondering and watching till we start to see little by little how they all fit together. So thank you, Carol, for that. And thanks all of you for having me.
And thank you, Margaret. That was lovely. The next person in line here is Dr. Tatiana Lobova. She spent 7 postdoctoral years at the New York Botanical Garden conducting research on bat-plant interactions in neotropical rainforest. Much of her fieldwork took place in French Guiana, often in the company of Carol and Scott Morey. Her bat-plant studies received funding from several federal and private conservation initiatives and have been featured in National Geographic Magazine, Bats Magazine, and on National Public Radio. Since 2007, Tatiana has been on the faculty of Old Dominion University in Norfolk, Virginia, where she currently holds a position of Master Lecturer and University Distinguished Teacher in the Department of Biological Sciences. Tatiana.
Good morning, everybody. I'm very happy to share my memories of a wonderful friend and mentor with you today. Next, please. I met Scott and Carol at a very young age of 28 when I came actually to the United States and came to the New York Botanical Garden, which seems like a long time and also a short time ago. And they were the people that influenced me the most since then, both professionally and personally. This is the photo of my last visit to New York. I was so overwhelmed and so happy to see them both. Unfortunately, the timing, as you can see, January 2020, worked out so well right before everything has closed for the pandemic. Next, please. Again, it's hard for me to separate what impact Carol had on me personally from professionally because it was so entangled. She taught me how to prepare for my first field trip in the neotropics and how to prepare Thanksgiving table for my first American Thanksgiving, how to take professional photos of plants, and how to be the best grandmother for the very active boys. Carol was a role model of graceful multitasking and organizing. And her organizing was exceptional and envious, at least for me. Next, please. This is a part of a tropical field trip hiking list that Carol made literally in the last century. And I was using all these years. Scott's grad students were using it as well. And now my students are also using it. So you can see how meticulous this parking list is. I had to adjust it. Of course, I had to make some changes for the times. I had to take out camera with films because nobody using it anymore. We went digital years ago. I had to add cell phone and chargers, but pretty much that's it. Only few changes been made. Everything else is still remain the same. It really is a perfect parking list. And I really wonder how many people out there are still using this list for field trips and eco tours. But you may be able to guess. One person that had never used this list for the field trips. That was Scott. And that's why every time we went to the field, he would always forget something. Next one. Guide to the Vascular Plants of Central French Guiana was the biggest tropical flora project that Scott and Carol did together. The first volume was published in 1997, and the second was published in 2002. I love this photo because they were so happy and because it's a typical outfit that we wore there every day, including the climbing spikes that Scott is holding and the ropes and the collecting poles. I was always dressed like Carol because she told me so, and Scott was always making fun of us because he thought we're overreacting and overdressing in too many layers, and he was the one that was getting constant diseases and parasites, and neither he or I ever got anything. So the joke was, of course, on him. The next one. I do not know how many times Carol actually went to French Guiana in over a decade times that they conducted this project, but I believe that they brought to Seoul, to Central French Guiana, more than 50 scientists and artists, not only botanists over this time. And both of them were extremely generous to share their friends and colleagues with others. For example, I became very good friend with Fanchon Prévost and Bernard Hermier in Cayenne because Scott and Carol were very good friends with them, and somehow it just spread on me. With so much knowledge and enthusiasm about nature, they have attracted so many charismatic and interesting people, both professional and amateur, and really enjoyed working with them. When I started working in French Guiana, Carol has happily instructed me how to organize the trips, how to make all the arrangements for flights and hotels, what Scott should not be eating, and she never went there again anymore. She was done. She wanted to move on and focus on ecotours from that point. Next. The last tropical plant projects that Scott and Carol have worked together on was in 2006-2007 with Bill Buck and Melissa Tulik. It was plants and lichens of Sabre. Sabre is a 5 square miles island in Dutch Antilles. All they told me, I never went there, it's beautiful and easy. Easy it is after French Guiana. Most of the photos of vascular and non-vascular plants were taken, of course, by Carol, and they're available now online at NYBG Sabre website, so you can see it there. Prior to that, Carol's photos were used in numerous publications, papers, and books, especially beautiful books like Flowering Plants of the Neotropics, published by NYBG, and our book, Seed Dispersal by Bats in the Neotropics. And I have samples of the pictures, Carol's pictures from that book here. Next. Besides taking beautiful and very informative photos of plants in the field, Carol, of course, was also collecting specimens. 1 genus and 8 species from 7 families were dedicated to her. Here they listed in alphabetical order, and I would like to read you this very kind and personal dedications that accompanied most of the species named, with the illustrations, beautiful line drawings by Bobby Angel. Next. We all were collecting under Scott's numbers, and sometimes that resulted in some tension between them, such as in this case, Rupert Barnaby's decision to name Dicyphenia mariorum, even though Carol was the one who found the flowers of the species on the ground. She was able to figure out that this is a new species, but Scott was the one that had to climb the tree and collect the specimen. So Rupert dedicated it to both of them, but he actually named it Mariorum. Next one. The Stiganthus graciae was named by Aguirre Santoro and Michelangelo after Carol Gracie, who photographed the plant in the field and also was part of the team that collected the type specimen. They also know that Carol Gracie was contributed invaluable photographs of plants in their native habitats throughout the Neotropics. Next. Marillo named both the genus and the new species after Carol and Scott, Gracie Mariana Graciae, because he was friends with Carol. He went to Saul, working in French Guiana, and he noted that she was the first to discover the plant and photograph it. She also collected and photographed many other species of Asclepiadaceae throughout the Amazon and Guianas. Next. This is a very nice dedication for Gustavia Graciae by Carnejo and Mori, who stated that the epithet of the species honors Carol Gracie, a North American naturalist who had photographed and written about plants and animals in Central and South America, Northeastern and Southeastern United States and Europe. Her striking photographs of plants and animals, as well as her writing about nature, has inspired many to love nature as much as she does. Next. Herae graciana was named by Bill Anderson, and he said that this is the most impressive plant, which to me means a lot because I think all Herae are actually beautiful. They have beautiful flowers, but he honored it and named it after Carol, an enthusiastic student of flora of Saul, whose excellent photographs are a joy to see and provide a valuable supplements to herbarium specimens. Next. Scott named Lesiris graciana and dedicated it to Carol because her keen eye discovered many interesting Lysiridaceae, and her fine photographs of them had made important contribution toward understanding the taxonomy of the family. I want to add that all of these authors have noted how important it is to have photographs accompany the specimens, because in many cases, when we get pressed and heat dry specimens, it's very hard to actually see the features, the micro features, especially, or the colors that are not preserved. So in all of these cases, Carol's photographs added very important information for describing the specimens and for describing the species. So that's why people, that's why all these authors, that's why all these botanists have to add that the photographs are very significant for description of the plants. Next. This is a very tiny plant, and I think without the photographs, it would be completely impossible to even describe it from the herbarium specimen. So Peperomia graciana was described by Aragors van Rijn, and she named this delicate tiny herb in honor of Carol, who has been extremely hospitable to all the scientists in Saul, and she also thanked her and Fanchon for providing this beautiful photograph. When this plant is pressed, it really looks like, pretty much nothing because it's so tiny. Next. Pachisia carolia scoriae was named by Marcano Berti and Aymart. They dedicated to both Scott and Carol, but they named it after Scott passed away. Next. In conclusion, I'd like to repeat how lucky I am that I got to work with both Scott and Carol, and I got to learn so much from them both. And they were so generous to share their friends, their colleagues, and their family with me. And over time, they became such an important part of my own family. And the older I become, the more I understand their influence on me. I also would like to thank the organization committee for inviting me and providing the illustrations, and to everybody in New York Botanical Garden for the great years I spent there and for the lifelong friendship that I formed with people that I worked there. Thank you.
Thank you, Tatiana. Thank you, Tatiana. Next, we will hear from Hella and Scott McVeigh. Hella McVeigh was born in Berlin and educated in mathematics at the Free University of Berlin. She taught mathematics for 25 years and was the founding chair of the mathematics department at the Stewart Country Day School in Princeton. She was the founder of the Whole Earth Center, the first organic food store in the Garden State, and a dedicated volunteer for Planned Parenthood, from which she twice received the Margaret Sanger Award. She is an explorer, photographer, painter, and writer. Scott McVeigh graduated from Princeton in English literature and began his career in the president's office there. Since then, he has been executive director of the Robert Sterling Clark Foundation and the Geraldine R. Dodge Foundation, president of the Chautauqua Institution, and member of the board of many organizations focused on arts, science, and education. While at the Dodge Foundation, he was instrumental in creating the biennial Dodge Poetry Festival, and more recently, he and Hella established the McVeigh Poetry Trail in Princeton. Having a long fascination with aquatic mammals, Scott has done research on dolphin cognition and has served as a US delegate to the International Whaling Commission. With Hella, he discovered and described the song of the humpback whale, which, as many of you may remember, became an anthem for whale conservation. He is the author of poems, scientific papers, and his memoir. The McVeighs are avid travelers, and together they came to know Carol on her eco tours to Trinidad and Italy. Welcome, Hella and Scott.
Okay, thank you for your introduction. It's over the top, but it was so exciting to hear Margaret's and Tatiana's presentation with those gorgeous slides and presenting the spirit of Carol and seeing that her name appears in a few discoveries. It's so exciting to me, but it's also very exciting to me that two lovely women are following her footsteps. That is so great, so wonderful to see. So when I want to talk a couple of things about Carol, I have to go back decades. The first time I saw her was in a field or meadow in the Pine Barrens. And she, I think, was looking at flowers, which became part of her Northeast famous book. She was traveling at that time with a local botanist. And it also was the time when there was pressure put on to preserve that very special part of New Jersey. So it was very exciting to me that such a famous botanist took interest in our little earth in New Jersey. And think about it now. When you fly from DC to Boston, when you look down on the earth, that place is the only dark place right now. Thank goodness we have some wonderful people continue the work in the Pinelands, and I want to mention two names, Emil DeVito and Carlton Montgomery, who do magic to keep that place in its gorgeous, precious state. My second time I spent with Carol was in Trinidad. I'm not a botanist. I studied mathematics, as it was mentioned, but my love for nature and plants and flowers was nurtured when I was very young by my spectacular grandmother to keep me away. As I mentioned, that I grew up in Berlin, Germany, in the worst of time. And the work my grandmother showed me in the gardens and nature kept me away from the evil and kept me sane. I guess it's the same way as people took walks and appreciated nature during the COVID. I bring this up because Carol had that magic my grandmother had, and she kind of brought that out in me. So in Trinidad, she organized a group of people who tried to sketch and look at the wildflowers. Carol knows and understood when you draw a plant, you don't just click. You have to look at it very closely, observe it. And that became a very precious, and look at all the precious details of the plants. Botanical illustration served through centuries before photography to understand what's going on this precious planet. And it still serves as a connection between science and the arts. And the poetry trail was mentioned earlier. And that is another way we have to connect people with all of our senses, the visual, music, poetry. Otherwise, I think this beautiful planet has no chance to survive. I don't want to take time up, so I will only mention briefly the third occasion was in the Dolomites. We took endless walks and hikes through fabulous territories, and precious things were pointed out all during the day. And then at night, after dinner, the folks of Kew Gardens argued forever and ever about the proper Latin word. That took me back to my Latin studies. I had no interest in that. So Cal and Scott said, oh, let's look at some plants more closely. Let's look into the sexy part, like, oops, and see the excitement of the flowers. And that somehow stays with me. And that's where all the excitement and the fun was found. Thank you so much.
Well, wow. Phew, thank you, Hella, for truly capturing the essence, spirit, joy, and grace of Carol. As a mere male, let me frame briefly our extended relationship with the New York Botanical Garden. After serving on July 12, 1988, another trustee and I from the W. Alton Jones Foundation called on Gillian Prance before he took on the leadership of Kew Gardens in England. And as we sought direction on our quest to protect biodiversity in the Amazon, Prance was vice president for research. He had led expeditions for 25 years to the Amazon to bring back 4 samples of every plant. Prance had published 7 books and written 130 scientific papers. I asked him if he planned to just keep on collecting or if he had an idea of what could be done to stop the wholesale depletion of the largest rainforest forest on Earth through cutting, burning, and mineral extraction. A light bulb appeared over his head. It led us to put $100,000 into a conference in Manaus with biologists and others from all the Amazonian countries, Europe, and the United States, and corporations. This is detailed in my book, Surprise Encounters with Artists and Scientists, Whales, and Other Living Things. And he's in there along the 152 other stories. But I just would also like to, even though every fabulous picture in the book has been held up, I'll just show a couple here. I'll put this up close to the camera to see if you can make that out. Yeah, you can maybe make out Carol and Hella. And then here's the other one. And this, you've got Carol right beside Scott. This is another one from Asa Wright. Nothing like the photographs you have shown all along the way. Hella held up her book that Carol Gracie gave us these books and asked us to draw. But okay.
Say something about
Yeah. So while Hella was drawing flowers in Trinidad, which she showed you and held up, I was writing poetry, and because they are such a team as shown in picture after picture, I'm gonna, from this book of 100 poems, I'm gonna read the one on Scott. The Sapucacia nut. All of life, from the chigger to the silver-beaked tanager, is a miracle. And yet, within that living, pulsing, vast net appear certain imponderables. A few imponderables like the eye, the platypus, the giraffe. Add to that list, near the top, two more: the Sapodocian nut, that huge glory from the canopy, the top of tops with its paradise or cream nut encased in shrunken amber, and Scott Morey's face, alit by the beauty of the nut and the joy of opening it. Margaret Roach did a terrific job of conveying Carol's impact on botany in the Northeast. Those three books from the Princeton University Press in 2220 and 2221, totaling 870 pages in the last 2 years of her life. The Princeton University Press, by the way, does superb field guides. Salute also to Tanya for reminding us of how many plants have been named, lovingly named for Carol, for species that she studied closely. Thank you very much.
Thank you very much, Hella and Scott. That was lovely to put a little bit of poetry in there. Now we can wind up. I'm sorry. Anyway, thank you, Helen and Scott. We end the program with Dr. Amy Burkhoff, a tropical ecologist who made a midlife career change from art to science, obtaining her BFA degree at the University of Colorado, Denver, and her PhD in biology at the City University of New York. Catalysts for this change included a tropical biology class at the New York Botanical Garden with Carol Gracie and Scott Morai, followed by one of their expeditions to French Guiana, where she subsequently spent a year conducting her thesis research. She is now wrapping up 21 years as a member of the biology faculty at the City College of New York, where she studies the ecology of neotropical wood-boring beetles, especially those associated with trees in the Brazil nut family. Amy, you're on.
Thank you so much, Jackie. And thanks also to Rob and Brian and Bobby for coordinating the program, and to Belisa and Ariel for helping with our technology today. So I first encountered Carol in 1989 when I applied to be a member of a volunteer-assisted expedition to French Guiana that a friend of mine had told me about, and I was promptly rejected. Next slide, please. But I was not deterred, and so I immediately signed up for a tropical botany class with Scott and Carol at the New York Botanical Garden Continuing Education Department. Next slide, please. So as soon as they realized that they had a class that included people with absolutely no prior botanical experience like me, they really rose to the occasion. And for Scott, this meant that he agreed to come in an hour early every week to coach us on our botanical lingo. But Carol, I think, was the one that really kept us coming back, because she was in charge of the tropical fruits. And so every week she would bring in fruits for us to eat that represented the families that we were going to talk about in class. Next slide, please. The following year, I was not rejected from the volunteer expedition. And so I ended up in Seoul with Scott and Carol. And they were both completely committed to our project and to tropical botany. But there was also this little division of labor going on where Scott was really always about the like, go, go, go adventure. But Carol made sure that there was always going to be some level of comfort and safety involved. So that was very much appreciated by people that went into the field with her. And in 1992, this was when I went back to school to start in the CUNY Plant Sciences program. Next slide, please. So I've often claimed that I followed in Scott's footsteps from a research perspective. And this is because not only did I follow him to French Guiana, but he also blazed the trail for me at working in southeastern Peru and finally in the Osa Peninsula of Costa Rica. But I think I really followed in Carol's footsteps as an educator, because without Carol's model of bringing in the tropical fruits, I probably might not have been going out every week before teaching botany class to the specialty stores, looking for bark, looking for seeds, looking for leaves from whatever plants we were about to discuss. And without Carol's model, I probably would not have cooked up ideas for extra credit projects like anybody who brings in a ginkgo seed snack for us to eat gets some extra credit. Next slide, please. When I started my job at City College, this was about the same time that Scott and Carol were wrapping up their work on the floras of French Guiana. And to me, this was a wonderful period for Carol because it really enabled her to delve into what I thought of as her true passion for native flora that really kept her occupied for her remaining years. And this was particularly valuable to me as an educator because I quickly realized that I was dealing with a student body where most of our students didn't know a plant from a hole in the wall. I mean, they come from all over the world. They speak almost as many languages are spoken in the UN at City College, but they don't have you know, a lot of history with our native flora. So it became a very important part of the class to me to actually take students outside looking at plants, and there was no better time to do that than the spring. So I actually started arranging my schedule so that I would always teach botany in the spring, because I loved having students watch that transformation from the winter world into the spring world. Next slide, please. So this really came to fruition about 10 or 12 years ago. And what happened then was that I inherited kind of the perfect urban wildflower walk. And I inherited this from my friend Jackie Bonavito, who was the one that originally told me about the trip to French Guiana. And she had inherited it from Bill Greiner, who used to lead this walk or other wildflower walks for the Torrey Botanical Society. Anyhow, it's perfect for car-free New York students because we could take the Metro North to Crestwood and walk all along the Bronx River up to Scarsdale and then take the train back. And just as I was starting to really like that as my spring walk, was when Carol published her spring wildflower natural history book. And this book was so wonderful to use with students because the chapters were just, you know, she scoured up the literature and transformed it into this very palatable chapter form for our students, not for our students, but our students took advantage of that. And the chapters were wonderful because they not only told you something about the history of the nomenclature and the morphology of the plant, but also about the interactions with pollinators and natural enemies. Next slide, please. So when I took students out on this walk, I was able to, you know in advance what we were likely to see and ask each of them to select one of the plants to help with the interpretation on the field trip, because I think it's really boring if you just have one person who keeps expounding the whole way. So I wanted everybody to participate. And those book chapters were the perfect way to do this. Next slide, please. And in the springs when I was not teaching botany, I would go with the members of my lab or friends of the lab or friends of mine from my local community garden. And it was kind of amazing to me to find out that even community gardeners that might plant spring ephemerals in the garden had never seen them free living. So that was just a wonderful way to get out there and start to look at the world waking up again. Next slide, please. Once the pandemic kicked in, I was especially grateful that Carol and her books had helped me pioneer this field trip approach to teaching botany, because while everybody else that I worked with was still pretty much stuck on Zoom, I was able to actually get out with students in a safe outdoors, well-ventilated environment and have these wonderful walks. And I really appreciated that. Next slide, please. I want to tell one story from her spring book. And this is the plant that I never let any of the students select because I wanted it for myself. And it's not my favorite spring wildflower, but I just think it has a wonderful, wonderful backstory. And her chapter, part of this was based on graduate student work published in 2004. And Frey had discovered, and I'm sure that most of you have noticed this yourself if you've ever gone out to look at spring wildflowers, that a lot of spring beauties, there's a lot of color variation within the populations, where there are individuals that are almost completely white and individuals that have these very, very dark pink nectar guides. And one of the things that determines this is the ratio of pigments in the flowers, with the white ones having more flavanol pigments and the ones with the pink nectar guides having more of the pinkish cyanidin pigments. Next slide, please. So it turns out that the pinker flowers tend to set more seed, which seems to imply that they are more attractive to pollinators, which we might expect. However, the pinker flowers are also more attractive to herbivorous slugs. And so you have these opposing selective forces on color. And it seems that the flavanol pigments actually deter the herbivores. Next slide, please. So why don't we just end up with a whole field of white spring beauties if slugs are an issue? Well, there's another natural enemy out there that participates in this spring dance, and that is a rust fungus, which apparently is preferentially associated with the white flowers. So you have this, this very complex dance of mutualists and antagonists going back and forth. And it's easy for students to maybe read in a textbook about, multiple selective forces and how they can have opposing selective, how they select for opposing regimes. But it's very, very different to actually go out and see these things in person. So I was really grateful for that synthesis. Next slide, please. So in 2018, I started this different scenario where I started ranging farther afield in the search of native flora and native hikes. And this really was timed for when Scott was no longer coming into the botanical garden. And Carol came up with this notion that we were going to have seasonal visits, which was a brilliant idea because, we get so caught up in our daily lives, and it was really very helpful to have that structure of knowing that we were going to try and get together once each season. It was totally manageable. Next slide, please. And she picked these amazing places. And this is where I really benefited from all of Carol's experience with tours, because she managed to pick all of these really interesting places for us to visit. They were all really special. They were all places that I had never been before, and they were all selected to be manageable given whatever health constraints were operating at the time. And like Tatiana, the last of the seasonal visits with the two of them was in January 2020, before the pandemic slowed us all down. Next slide, please. And finally, I wanted to end with what is next. And for me, I think one of the things that is next is to go back to some of the literature. I know there's at least one other graduate student that has worked on spring beauties after the publication of the spring book. And since I no longer have Carol to do all the synthesis for me, I'm going to go back and look up and find out whatever subsequent research has been published on spring beauty since then. And I'm also really planning to make a deeper dive this year into the summer wildflowers. The summer wildflowers was published in 2020, and I've kind of done some pecking around in it, but I haven't really done a deep dive yet. And to me, the spring and the summer books are, for me, the most wonderful part of Carol's legacy, because they are things that I can use on a regular basis that make me feel closer to Carol. And so if anybody that is out there today hasn't yet enjoyed these books, I really encourage you, because it will be, if you use these books as your guide, it will be like going out for a hike with Carol. So that is it. And thanks to friends and family that are here today.
Thank you, Dr. Amy, for our science lesson. We have reached,
Very welcome.
although we have reached the end of the program, this Zoom session will be live for an additional 20 minutes. We won't be here, but the session will be live. and any audience member can stay in session and use the Q&A feature to submit any personal comments or memories about Carol that they wish to share. I send heartfelt thank yous to each of our illustrious speakers for their very enjoyable stories, and to all attendees for taking time to be with us to celebrate Carol Gracie's life. Remember that feeling sad about losing Carol is the natural result of feeling so glad to have had her in our lives. Thank you for coming and have a good afternoon.